Hello, this is Joseph Anthony Campagna. I'm a master's student in bioinformatics at Temple University, and today I will be discussing my thesis, in silico rational design of DNA origami-based allosteric metamaterials. Okay, so there is a design problem in molecular nanotech or DNA origami. Essentially, how do you create structures that respond to environmental signals? And we have, programmable structures like DNA origami, but they lack the sort of dynamical control that something like enzyme complexes have, unless guided by rational design principles. So, a key question we can ask and keep in the back of our heads through this talk is how can we engineer collective conformational changes, like signal relays in DNA origami structures? And can we look to Mother Nature for inspiration? So, it turns out we can, in one great example that is bacterial chemotaxis. You have a dimer in the left of figure A, which is comprised of chemoreceptors A transferring a phosphoryl group to chemoreceptor Y to mediate or modulate motor functionality, as well as several other evolutionary conserved chemoreceptors. And then what happens is this dimer aggregates with two other dimers in some well-investigated models of this phenomenon and forms a trimer of dimers. Finally, you get a core signaling complex that's not studied at the basis of the periplasm in such a way that is, maximizes surface area, but actually aggregates as a core signaling complex within a single region of the system. So what's interesting here is we don't know exactly how or why this works. And we have, in this research in particular, someone or a group of researchers who takes a set of parameters, in this case, receptor length in angstroms, and maps it as a function of gamma or the glycine hinge deflection. And what they find is that there's a region of acceptable conformations for this, dimer complex versus forbidden areas where there would be clashes between trimers. So this is sort of inspiration for what we'll be getting into. Now, from enzyme to structure allostery. Allostery, of course, is regulation at a distance where you have input at one site causing structural change at another. Allosteric proteins such as hemoglobin inspire a design paradigm for signal-induced bulk conformational change as well. And we hypothesize here that DNA origami can be designed with mechanical allosteric behavior. So towards allosteric metamaterials as opposed to allosteric proteins. We're going to consider that they're DNA origami based, so built from scaffold and staple strands, where the staples are guided by the scaffold in vitro to create molecular design. We say that they're allosteric, meaning they're capable of signal propagation via conformational change. We say that they're a metamaterial, meaning that their collective properties arise from designed internal architecture. And these systems are not static. They change form, flexibility, or function in response to stimuli. So you have some Japanese researchers here who investigated a similarly used cross-shaped DNA origami lattice that experiences adsorption rather to a lipid adhesive. And what happens is when you affect something here at the left and leftmost end, it will, in principle, affect something on the rightmost end, and that is allostery. So our design objective is to create rationally designed hinge linker architectures whose bulk conformation can shift in response to input signals. When I say bulk conformation, I mean having several DNA origami, whereas I'm modeling a singular DNA origami for in multiple different conformations. So properties we seek are responsiveness, tunable flexibility, and propagated conformational shifts when treated as a bulk signaling array. Analogous to membrane spanning receptor lattices in bacteria when treated as a bulk array of cross-shaped origami, going back to chemotaxis. So, what controls flexibility? Just like the researchers in the chemotaxis study looked at receptor length, for example, and some other parameters that escape me right now, which parameters in our case determine the hinge mechanics? And do we have flexure zones that can be encoded? In our case, this would entail investigating various linker lengths of the hinge. So in my case, in my thesis, I had done CAD modeling with CAD Nano and OxView, primarily OxView. I then simulated using the LAMPS OxDNA2 bridge, where I built an input file that essentially had taken into consideration parameters for, biological parameters in terms of buffer salt concentration and uh, time step of about, I believe, 10 nanoseconds, if I'm not mistaken. We analyze angular distributions, end-to-end distances, and correlated movements. Um, or at least this is what we'll get to further. The research is not complete. We did analyze angular distributions and end-to-end distances. And tools for this were OxDNA, VMD, and custom bond angle metrics. So if this video will work, here is a video of one of my simulations. This is the 20 linker, meaning that blue beam you see in between the two larger beams is the hinge of 20 nucleotide base pairs, whereas I have one of 10, and another one of 30, and another one of 40, as well as the 20 linker. Okay, how do I get out of here? Okay, so now this is my results. My results are we have a standard deviation of bond angles over frames, where we have the bond angle on the y-axis and the simulation time on the x-axis. And what we notice may not be clear from the colored lines, but when we take the mean standard deviation of each of the distributions, the angle distributions, and what this is the angle between that hinge and the lower beam. So you try to imagine ideally it's a 90-degree angle, but in reality it's a little messier than that. Essentially, what I've discovered, assuming all things else are correct, is that the nucleotide linker length um, becomes, we see more stability with a greater nucleotide linker length. We see less flexibility and hence more rigidity with a longer nucleotide linker length, which is counterintuitive. You would expect to see, the 10 and 20 to have this higher or this lower standard deviation in bond angle distribution, when in reality, it's the opposite, or at least computationally. So, summary and future directions. We introduce a framework for allosteric mechanical metamaterials based on DNA origami, inspired by natural collective systems like chemotaxis arrays. Future directions: quantify signal propagation length, explore feedback loops within allostery, integrate ligand binding modules, and build multi-layered decision-making nanostructures such as DNA logic gates like the Douglas lab does in, I believe, ASU. It's very fascinating stuff that would, you know, more research like this would give rise to essentially better nanorobotics in the future and a revolution in medicine with precision nanomedicine. Thank you for your time.